All right, next we're going to be hearing from Angelo Poletti.
Okay. I hope you will understand me in my Italian accent. Trying to speak slowly. This is a pretty, well, first of all, thanks for giving me the opportunity to present the data here. I only done work on self-model, so there is no direct translation to clinical studies yet, but I hope that some of these results can be applied in mouse. This is a cartoon pretty much similar to the one that Kurt has already shown before. And the message is pretty much the same.
Is the switch on the side on PowerPoint? On the side of the you should say PC.
It's okay. So the cartoon is pretty much the same that Kurt has already shown you, and the message is pretty much similar. The mutant androgen receptor is activated by testosterone, and this induces the formation of aberrant conformation proteins that are thought to be participating inside the cells. So the red arrow indicated that there is a detrimental effect of this aberrant conformation inside the cells. And there are many studies have been demonstrated that something can go wrong inside the cells because of the presence of the neurodegenerative receptors. At the same time, the cells activated some pathways that are beneficial to the normal response of the cells to the mutant receptor, to the aberrant conformation of the mutant protein inside the cells. So we may have different way to approach this disease. We can try to counteract the activation of the aberrant conformation by counteracting testosterone here. Well, we can use this as a pointer. Or, as has been shown this morning, we can try to counteract the effect of all the species inside the cells working against the red arrow. Or we can try to potentiate the positive pathway inside the cells. And among this, we know that there are molecular chaperones, and the two major development pathways that are the proteasome system and the autophagy system, they work in this direction. They try to remove the aberrant species formed inside the cells. So the idea is that when testosterone induces dissociation from the tissue proteins, there's a crucial step here that induces the formation of this aberrant conformation, because the receptor has to change in this folding. And because this requires for its activity and to direct the receptor into the neuron. So at this point, this aberrant conformation can be recognized by the proteasome system in one direction, or to doesn't matter, to the autophagic system. And there is a tendency of the misfolded proteins, the mutant androgen receptor, to collapse together into the aggregates, and at least at the beginning, this seems to be a sort of protective mechanism waiting for the degradation by the autophagic pathway. So there's a very nice equilibrium inside the cells, and everything works well until something goes wrong, and maybe a lower level of one of the chaperones, a reduced activity of the proteasome system or the autophagic system. And what is emerging is that there may be a problem with the clearance of the mutant proteins by the autophagic system. There are several data showing that the autophagic system cannot efficiently remove these species. A lot of work has been done from several other people working on SBMA, like Edelmanti and the American in this field. And we believe that if we can facilitate in some way the respiration of correct autophagic flux, we may facilitate the removal of these misfolded proteins from the cells, and it can be beneficial. So the question is, how can we improve this solubility and degradation of the mutant proteins? So I mentioned the chaperones. There are several chaperones, and they have different roles inside the cells. There are the HSP70 proteins, HSP90 proteins. They are there to refold the misfolded proteins, but there are other chaperones, small itch-up proteins, that directly recognize the mutant protein with a valid conformation, and then make a decision, a choice. They can assist the other chaperones for refolding, like in this situation, or they can direct the degradation to the proteasome, and now we know, there are some recent data that we have, that can also facilitate the autophagic process. Among all these small itch-up proteins, there are 10. 4 of these are expressed in the brain, but only a few have been found in some way involved in the recognition of misfolded proteins and in the activation of autophagy or facilitation of autophagy. And one of these is HSPB8. This particular protein is also mutated in another motor neuron disorder, a Charcot-Marie-Tooth disorder, which has similar feature with SBMA. So we focus our attention on HSPB8, and we studied if HSPB8 can be over-induced in the motor neuron cells of another motor neuron disorder, amyotrophic lateral sclerosis. And what it turned out is in the motor neurons that survive at that stage of the disease, that still express large amount of SOD1. There's a very, very high expression of this protein, HSPB8. And we believe, or we'd like to believe, that those motor neurons survive at that stage of the disease because of the presence of this chaperone. We look at the expression in ND mice. We didn't see actually an increase in the spinal cord, but the effect can be diluted because the number of motor neurons present in the spinal cord is very low. But there is a very, very high expression induced by the mutant proteins in the muscle. So the muscle responds to the presence of the mutant proteins by increasing the level of this chaperone. Is this chaperone capable to remove the insoluble species? You can clearly see here that the androgen receptor accumulated after induction with testosterone. And if you overexpress HSPB8, you completely remove all these insoluble species from the cells. It's in some way activated the clearance of the receptor from the cells, the number of aggregates decrease, and this occurs without altering any other pathway inside the cells. It does not interfere with the proteasome system, but we know that if we block, we drug the autophagic system, we lose the effect of HSPB8. So HSPB8 acts by facilitating the removal of these species by autophagy. And we have other data proving this can be improved. So the problem is now we cannot use proteins, and this protein, it's hard to test in the animal model because unfortunately it doesn't work on the androgen receptor with a very, very long polyQ sites that are being used, produced in mice model that are available now for SBMA. But we know that it works in other misfolded proteins, like SOD1 and TDP43, which are responsible for ALS, in Huntington, for Huntington disease, ataxin-3 for spinal cerebellar ataxia. So if we cannot test these proteins in SBMA, we can test this in other neurodegenerative disorders and hope to translate the results in SBMA. So this is our idea now. In some way, HSPB8 restores the autophagic flux inside the cells and eliminated the misfolded species, so prevented this accumulation and probably all the other toxic mechanisms. But it is a protein, it is hard to administer proteins to humans, so we have to find a way to induce the expression of HSPB8 directly in motor neurons with drug. So we set up a system in which we can screen for a large number of drugs using the region that control the expression of HSPB8 in human. We produce several models, and we are doing now a robotic screening of NIH library of commercially available drug and natural compound. And we just started, and we will have the results soon. But some of the drug in which we were very interested were sex hormone steroids for several reasons. And so we tested directly the action of these hormone steroids on the expression of HSPB8. And the first was androgens, testosterone and DHT. And we were surprised to see that actually testosterone and DHT reduced the level of HSPB8 inside the cells. So it seems that if we activate the androgen receptor inside the cells, we reduce the level of HSPB8, so the cells respond less to the presence of misfolded species. If we use estrogens, that actually are derived from androgens, and are the hormone that are present in females, we will see a very high increase in the expression level of HSPB8. Actually, it's enough to express the estrogen receptor, so there may be a ligand-independent activation. But if we use also androgens, the level of these, let's say, protective proteins increase very much. And this may explain some difference in the progression rate and the risk to develop ALS in male and females. We cannot say if this is also involved in SBMA, because in SBMA we know that testosterone is a trigger of toxicity for several other mechanisms, but can be a contributing factor also in SBMA. So they can be a good candidate because there are several compounds that have androgenic estrogenic activity, and we can take advantage of this equilibrium if we can block the inhibition induced by androgens on the expression of these protective proteins, or we can enhance the activity of estrogens in inducing the expression of HSPB8. And there are several drugs, and some can be used are anti-androgens, like the casodex and cyproterone acetate. There are also natural compounds that we are testing to see if we counteract the inhibition induced by testosterone, we can increase the level of HSPB8 in our motor neuronal system. But also we have estrogens. There are several estrogens that have been developed, selective estrogens modulators like tamoxifen, raloxifene. It can be a mixed estrogenic or anti-estrogenic compound depending upon the cells. We tested the activity of tamoxifen and raloxifene, and they act as estrogens inside the motor neurons, so they are able to induce the expression of HSPB8 in our model. And those have to be tested in mice, but can be a possibility because they are already commercially available, and we know a lot about these two compounds that have been used in breast cancer. And there are also some natural compounds that seems to mimic the activity of estradiol on the HSPB8 promoters. So genistein, for example, induced the expression of HSPB8 in our cell model. So it would be interesting to test the effect of this compound also in the mice. And the last one that we tested was trialose, because trialose was already reported to be an inducer of autophagy. And so we wanted to see if in the mechanism of action of trialose, HSPB8 was involved, and actually trialose is not only an inducer of autophagy, but also an inducer of the expression of HSPB8, which is not an activator of autophagy, but a facilitator of autophagy. So it could be a mediator of trialose activity, and trialose are already being tested in several animal models to see if there is any protective activity of trialose. It seems to increase the survival of some of these animal models for neurodegenerative disease. And actually, a combination of trialose and HSPB8 is even more active in the removal of misfolded species inside the cells. So I think I can stop here. So I can, if you want, I can take some questions.